Welcome to today's webinar, Seeing the Whole Blueprint, Uncovering the Purpose of Genomic Junk. I'm Ankur Jain. I'm a Whitehead Institute member and an assistant professor of biology at MIT, and I'll be your host and moderator for today's event. Today's webinar is the second in the series of talks that we have organized at the Whitehead Institute to share our science with the broader community. Our topic today is Seeing the Whole Blueprint, Uncovering the Purpose of Genomic Junk. And in a few moments, we'll hear from our presenter, Whitehead Institute member, Yukiko Yamashita. She'll highlight the ways in which fundamental research that is being done at Whitehead brings novel biological processes to light. And she'll introduce us to her lab's latest research on the function of satellite DNA, which had long been regarded as genomic junk. Before we get started, a couple of logistical reminders. This webinar is being recorded for future viewing. Yukiko will also be fielding questions towards the end of her talk, and you can submit your questions at any point by using the Q&A box. With that, I would like to introduce Yukiko Yamashita. Yukiko is the Susan Lindquist Chair for Women in Science at Whitehead. She is a professor of biology at MIT and an investigator with the Harvard Hughes Medical Institute. The Yamashita lab studies the process of germ cell development, a process that is critical to the continuation of species. Yukiko earned both her bachelor's in biology and her PhD in biophysics from Kyoto University in Japan. She conducted her postdoctoral research at Stanford University, and prior to joining us at Whitehead, she was a faculty member at the University of Michigan. Yukiko has earned numerous very prestigious awards, including being named a MacArthur Fellow, a Searle Scholar, and she, as well as grants from Keck Foundation, Niagara University, and the American Society for Cell Biology. With that, I'll hand it over to Yukiko. We are looking forward to learning about your work.
Thank you very much. I have to share the screen first, and then this way, and then get it. So is this okay? Yeah, thank you very much, Anka, for the introduction. And I'm really, hi everyone. And I'm really excited to present, what my lab is doing right now. And kind of, yeah, and then, well, so I want to give you some sort of, the Southern view, more than the detail of the data or that kind of things, and especially like how to say, my hope today is to convey this, excitement of exploring into really uncharted territory. And hopefully you can see that, that you can't really prescribe what you might discover. You have to just go with it, and but that gives you so much, the joy along the way. So today, I want to really tell you about, function of junk. What is it, right? So I think, the one, why I decided to study this is I often, one sort of misperception I encounter often is this. With the genome sequences, we have human genome sequence. Many people feel the world is like this. We have blueprint of everything, every single tool, and we know how it works, etc. So that means with the CRISPR technology, for example, these days, if you find any mutations in any patient, now you can fix it right away because we know exactly how it works. But I'd like to say that's actually the big misperception. Because, if you look at all those, the blueprint of our, the genome, often case we find something like this, something that looks like a tool, but you don't know what it is at all, right? And then if you find, that you find a mutation or some sort of variation in that kind of tools of unknown function, we have a, we as a, as a whole community, as a society, or, the medical field, we will have a harder time to really see if that is really causing certain, the symptom of the disease, or it's just a harmless variation. So you start with this, whatever the tool of unknown function. Let's say you find a variation in a patient, something like this. Is it bad or it's okay? or something like this, the things are, in half. Is it bad or, it's okay? Or it comes in a different color. Is it good or bad? It's really difficult to judge, which is really in contrast to the situation where we know the function of the tool perfectly well, such as screwdriver. So in this, in case of screwdriver, we know exactly how it works. That means if you see a mutation, something like this, the tip of the screwdriver is chopped off, we know it's not going to work. So that means we have to fix it, and then we probably know how to fix it. But because we also know how screwdriver works, it comes in a variation in the color of the handle or a little bit of slight difference in the shape of the handle. We know this works. So this variation is not going to affect anything. So now let's go back to this mysterious tool. How can we judge any variation is bad or good? So for that, you need to understand the mechanism of action, how this is really functioning. And then to just give you the answer for this particular tool, this is how it works. Actually, you can actually pop it up. And this is actually the portable, coffee dripper when you want to bring your coffee to, the outdoor activities like camping. So now, finally, when you understand the mechanism of that tool, how that tool works, now you can tell the color variation, this blue one, is totally okay. It's not gonna, interfere any of the functions of coffee dripper. If it's in half, probably it's not gonna work. You have to find a way to fix it if you wanna have coffee. If you have a big hole in the middle, but now that you know what it is, probably if you can put the, coffee filter or maybe two of that, and then if you know to, the slowly pour the hot water, probably this is workable, right? So this is one thing. And then, in studying the all sorts of, the going into the mechanism of, how life works, we use Drosophila in my lab. And then, if you look at this fruit fly, often case, I also get a lots of questions. Why do you study this tiny insect? That is that really useful at all? The Drosophila was established as a model system by, this famous Thomas Hunt Morgan. Without him and his colleagues' contribution to the field, we wouldn't know that all those genes, which is an element of the inheritance, are on the chromosomes. Also, many genes involved in cancer were first identified in the Drosophila. So, the demonstrating the importance of the model system. So my view of using Drosophila is something like this. To understand, how, the Lego house is built, you can start with a little bit of, you want to start with a little bit of simplified version of it, something like this, before you can go into way much more complicated Lego castle. Really important aspect of it is, both of these, the tiny Lego house or ginormous Lego castle are both made of the same unit, such as cell. So by understanding smaller version, now I think, you can actually get the much, the better picture of how more complicated things might function. Okay, so I just told you that our genome is full of tools of unknown functions, right? That sounds bad enough. But if you look at the actual whole genome of our human chromosomes, things are even worse, because all those tools of known function and unknown function only goes to 20% of our genome. So the remaining 80% are mostly considered as junk. What does that mean, right? Okay, so let me explain what is junk. So this is the the micrograph version as well as cartoon version of the chromosome on which our genes are on. These are made of the DNA and the protein complex, and then that contains all of the blueprint of our life. So, and then also the genes typically that the code for the blueprint for, or the recipe for the protein or tools, right, are typically on this blue, the light blue area. But you see ton of, the area that doesn't have any, the so-called coding capacity. So these, sorry about that, these area often occupied by so-called satellite DNA, which is a tandem repetitive DNA. For example, AATAT sequence is repeated a million times for megabases of the DNA sequences. And these, the satellite DNA is really ubiquitous and abundant, can be more than 50% of the genome, depending on the species. Despite this abundance of ubiquity, this has been typically regarded as junk because it's non-coding, meaning it doesn't produce any proteins. So if you look at the, the language of the DNA, it really looks like a gibberish. So that's why this, the satellite DNA has been regarded as, or disregarded as junk. So several years ago, I and my former postdoc, Madhav Jagannathan, when he started, we sat down and they said, it doesn't make any sense. 80% of our genome is completely junk or useless. That's how we decided to study this. And that was the only, can't be completely useless. That was the only motivation for us to start this. So then since then, what we discovered is this. So in the, this is a typically, any, cells contains the multiple chromosomes, right? But then, these chromosomes as a shape only appears in, the only certain phase of the, when cells are only getting ready for the cell division. But in the real life, these chromosomes are more, doesn't have a shape and then just packaged in the nucleus. And when that's happening, people have known this satellite DNA kind of associated with each other from the multiple chromosomes. And this aggregation or clustering has been cytologically defined as a chromocenter. This presence of chromocenter as a structure, cytological structure, has been noted like almost, the 100 years. But its function has not been known. So what we found is, sorry, and this is actually the real version of the chromosome you can see over here. And that this blue shows all those chromosomes, that when cells are getting ready for the cell division, and these magenta or red decorating the chromosomes, these are the specific satellite DNA sequence, in this case, AATAT repeats. And if you look at on the individual chromosome, you see many spots, right? But in a real cell, you see the same, this, the red sequence comes into one spot like this, the over here. So this, that represent this, the clustering of the chromosomes. And we discovered how this is, this chromocenter is formed. We found a protein called D1. So, and then this protein has the ability to bind to this AATAT sequence and then bring multiple AATAT bearing DNA together to, to bundle many chromosomes, just like a Velcro. So, and then, as you can see here, DNA strand is over here, and then D1 can just bridge them together. So that means in the absence of D1 protein, you cannot bridge the chromosomes together. And then this is what you see. Over here. Now, instead of having one major, the clustering over here of the AATAT, D1 mutant, which cannot cluster this DNA, AATAT DNA, have multiple AATAT spots like this. Okay, so then the next obvious question is, what do chromocenters have to, why chromosome has to be bundled, or what's the function of the chromocenter, right? And then if you mutate these D1 proteins to disrupt the chromocenter formation or chromosome bundling, you lose the cells a lot. So this is a control situation. This Drosophila testis contains lots of germ cells, which is a precursor for making sperm. And the control or wild type situation, you have many cells showing green over here, but the mutants quickly lose the cells. So that means, at least that told us, this protein or chromosome bundling is important, right? Because cells are dying. But why cells are, how cells are dying? Without knowing it, the only conclusion we can make is, yeah, this gene is important, period. So, but we are kind of, stuck over here. Like this is important, but how this is important, why this is important, we didn't know. And, we had, that this is the research, unprescribed research. We was really stuck about a year, and we don't understand why this is happening, how this is happening. And then, Madhav and I was, staring at the, this image for a while, actually during our meeting, I believe. And then we were just staring at it. And then one moment we realized, what is this? I don't know if you notice, here's a black spot, which is not green, right? So, and then we started wondering, what is this? Why there is some sort of, the dark holes over here? What does this mean? And then when we saw this, now we thought we might have an answer, which turned out to be the case. So it turned out to be, it's a small nucleus. In control situation, each cell have just one nice round nucleus, which is shown in the green over here. All DNAs, all of your chromosomes are, they encapsulated one nucleus by one envelope like this, in a circle nucleus. If you don't bundle the chromosome, what you see is some chromosomes start coming out of this nucleus to form its own tiny nucleus like this. And it turned out this was a reason why cells are dying, right? Based on these data and some other, a little bit more detailed data that I'm not going to show. So we came to this, idea. Okay, so the chromosomes are typically bundled by chromocenter, like this. And then, as I told you, to bundle all those chromosomes, you need this, the protein that can, to bring all the DNA together by binding to saturated DNA, which is the D1. And this bundling of all those chromosomes is likely the reason how cells can encapsulate everyone in one nuclear envelope. In the absence of this bundling, now individual chromosomes are free from each other. They are not anchored to each other. So now individual chromosomes start floating out of the nucleus to form its own micronuclei or nucleus. So then this actually leads to the cell death. Okay, so that based on that, we want to really propose junk or saturated DNA is actually not really junk. It's a critical constituent of the chromosomes to form a single nucleus. So let me just recap as conceptually. So we human beings have 46 chromosomes, right? And that all, that together, they encode the whole blueprint of our life. So that means all the books or all those blueprint has to be in one place, which is that nucleus. Everything, all those information has to be in one place. This is very much like the situation where you have to put all the volumes of encyclopedia in one place to have a full information. So then in case of the chromosomes, this, the putting all information in one place is done by physical linking of the chromosome, all those books together, just like a bookshelf, then a nuclear envelope, does encapsulate all those chromosomes. And this bundling or, the gluing is done by a protein called D1, just as I told you. Okay, so that means, right, the people thought, oh, saturated DNA does not have its, the content information. If you go through the page of encyclopedia, this saturated DNA does not participate to that part. Instead, it doesn't have a content information, but it's critical to contain the information, very much like a book spine or, so that which books to put together into one place. So now, in the last few minutes, I want to spend just a little bit of a time, how we are going with this information. And then again, I want to kind of illustrate how, the prescribed research might go. So finally, when we convinced ourselves that the saturated DNA is not junk, we had to face one interesting, well-known fact, which is, the saturated DNA is actually highly divergent between even very closely related species. So that is true between, for example, human beings and chimpanzee or gorilla. the coding sequence or, the non-junk part of the DNA is very much similar, but a satellite DNA is very much different between those two species. Same things, true with Drosophila melanogaster. That's our laboratory, the model organisms. And its sibling species called the Drosophila similans. And all those different satellite DNA is shown in a different color over here on the chromosome. And then you can just, you don't have to know what it is, but just the color difference tells you melanogaster and the similans don't have any, don't have much similarity. But as an insect, they look very much the same. And if somebody bring one fly to me, is this melanogaster or similans? I won't be able to tell. The melanogaster and the similans are very much alike, and then they, the most of the genes are very much the same. So, but now, okay, let's think about this. Those two species have very different satellite DNA, like depicted in the blue or greenish satellite DNA versus orange satellite DNA. If you make a species hybrid, meaning you mate the Drosophila melanogaster with similans, so they are that close, they can mate, they are that close, but then they bring in two kind of chromosomes together, right? This is very much the situation when you have one set of encyclopedia and you have a completely different second set of encyclopedia. Their contents, if you probably go through, each entry, each contents of these two encyclopedia, I'm sure you can find a very similar information. Contents is very much the same, no matter which country you live, our knowledge and, all those, things are pretty much the same, but they are those, the information are bundled slightly differently. So that means they are not exactly the same. So is this a problem or not? So that it became our idea. So, okay, so you can cross, as I said, melanogaster with similans. If you do so, the hybrid, the progeny of this crossing would be, the hybrid female, the daughters are sterile and then sons are dead. And then if you look at this sterile female, they barely have any germ cells, that's egg precursor cells in their ovary. So this is how it actually looks like. And over here, I'm showing melanogaster, just a pure species ovary. And then these blue cells over here, like there's probably tens, 30, 40s of those cells over here. This is a precursor to generate the eggs of this melanogaster, right? On the contrary, if you make a hybrid, they still make an ovary fairly okay, but these blue cells, these egg cell precursors are very much reduced. And then eventually by the time this, the ovary fully develops, all those germ cells are pretty much gone. Okay. And then, but we wanted to take a look even a little more, the closer look. So, okay, so here's the idea, right? If you look at the pure species, all nucleus contain all the chromosomes with the compatible satellite DNA like this. So that means these chromosomes can be bundled completely okay. That helps to contain all information, all the volumes of the encyclopedia into single nucleus. That's how you see very nice, the nuclear envelope that's encapsulating all the DNA in it. Here is a situation. It's a pure species, but you have the protein, the bundling protein mutated. In that case, you cannot bundle the chromosomes. Then you end up with having micronuclei because now all those books are scattered. So that's how you see the micronucleus over here, right? Now in a situation of a hybrid, now you have a two set of the books that is not quite compatible, which we thought. So maybe these, the chromosomes with the same satellite DNA might be bundled. We don't know, but we guessed. And then, these orange satellite DNA containing chromosome may be bundled okay. But can they bundle together or not? That became a question. And indeed, as we suspected, in the hybrid situations, those the germ cells, egg precursor cells have ton of micronuclei like this, that there's a big nucleus and there's some another nucleus over here and then smaller nucleus like this, meaning, or, to us, this suggests that, that they fail to bundle chromosomes as this cartoon indicates. Okay, so now I think, that we are coming up to this idea. This might be a reason why hybrid cells are not happy. And then, the chromosome from two species are not quite compatible with each other. And then we wonder, this might be how, the species split into two. Just to summarize, that today, that I told you about the satellite DNA, which has been regarded as junk, but our conclusion is it's not junk. It doesn't contain contents information, but it has a critical function to contain the contents, right? To bundle the chromosomes, right? Connecting all those chromosomes for the encapsulation, to put all those volumes of the books into one place together. So that's the function we think. And then, the second part, now we are guessing this, the divergence in the book spine or those kind of meta information, not the contents, might be the cause of the hybrid incompatibility or how two species get split from each other. With that, and then, the actually knowing satellite DNA is not junk has opened up so many lines of the inquiries in my lab. And then so we are really excited in addressing those, the really new questions that, came out of this, the finding. So with that, I want to thank all my lab. And then, I just joined in the middle of the pandemic. So that means we actually never get to have a chance to have a group picture yet. So this is a, patchwork of the old lab and the new lab and so on. But, I can't wait to, for the day that everybody get together and to take a new picture. And I want to thank all of those, for the support, for funding, and then community, and so on. And thank you so much, and happy to take any questions you might have. Thank you.
Thanks, Yukiko, for that wonderful talk. It's amazing. We have several questions pouring in. I'll start with the first one. One attendee is asking about what technologies do you use to visualize these repeats, and in general to conduct this research in repetitive elements, and what are the key limitations of these technologies?
Right. Yes, so just to visualize those specific DNA on the chromosome, it's relatively easy. That's so-called DNA in situ hybridization. So if you have, the one strand of the DNA, you can design the complementary DNA that's going to hybridize with each other. I mean, they, adhere to each other, they form the DNA double strand. So in doing so, you can design a probe that is, the bound with a fluorophore, so that, the AATAT can be visualized in red, and then some other different sequence can be visualized in green. So that is, the visualization is not that bad. The really challenging part is this repetitiveness of a sequence. That means it's really difficult to completely read those sequencing and then align on the chromosomes. So when we typically say, whole genome sequencing, we typically just ignore repetitive region. So that means even today, the entire sequence of the repetitive region, including satellite DNA, is not really completed for most of the species. For some, the leader species, we started having some, the pretty good assembly of exactly how repeat region might look like, but it's not complete, and then far from knowing of all different species yet. So that means, it's really difficult to, if you don't know what's in there, if you can't sequence, it's really difficult to go after, right? So and then we often joke, that means, that there's no competitor, nobody, because they don't have information. The problem is we don't have information either. So it's still a challenge, but I think, So my hope is, that by doing, our kind of research too, we can hopefully get enough number of the people get excited about this, and then more effort is put into sequencing and then really mapping these, the satellite DNA of the, of the many species genome. Yeah.
Another question is, what portion, so junk DNA is about half of the genome, as you mentioned, what fraction of it is responsible for chromosomal bundling? And are there any other functions that you hypothesize or that your lab is looking into?
Yeah. That's a really interesting question. We don't know how much is necessary or how little is sufficient. And that is also related to this difficulty of, first of all, difficulty of reading and completely assembling the sequence information. And without that, it's really difficult to manipulate those sequence. So for example, now, that we have so much technology of editing the DNA, but if that's the target is a unique sequence, you can precisely go to that target sequence to manipulate whatever the way you like. But if you have same sequence repeated over and over and over again like this, you just cannot manipulate this repetitive DNA so easily. So I think that we don't know the exact the degree of how much of the satellite DNA is required to do the job that I described today. With that said, we do have that we find the satellite DNA in other weird places, such as in intron. We have been studying some really interesting, gene structure. So that is, the so-called coding sequence is like about 10 kilobase, but it contains an intron that spans like megabases, right? It's so much bigger than the actual coding sequence. So it seems like this, the ginormous intron seems to have some function in a gene regulation. We don't know exactly what it is, but we started, the tackling on that kind of question. So, the satellite DNA seems to be everywhere, and exactly how they function. I mean, just so much to explore, and then so little we know about them. Yeah.
Related to that, how do different species know that they have to evolve different repeats, and how do they do that?
Yeah. So I think this is more like, they are not purposefully evolving different the protein or different satellite DNA. So by the nature of the repetitive DNA, so they are known to be very unstable, meaning one time you might have 1 million AAT repeats. It's so easy to lose some of them. Let's say it becomes 500,000. But then because you need to maintain some amount of saturated DNA because it's functional, it's not junk. So that means, based on the knowledge in the field, I think it's quite conceivable. Whatever the saturated DNA has this fluctuation of the shrinkage and the contraction and the expansion, contraction, expansion. And then this is just for the sake of maintaining satellite DNA amount, right? But then if you when you are expanding, there is so much room to introduce a mutation. So one time you might have AAT repeats. When you are expanding, you made a mistake and some of the repeats become AATC instead of AATAT, probably that is well tolerated by the cell. But if you keep doing that, and the one day you had 90% AATAT, only 10% AATSC, and then whatever the years later, this percentage might flip. When that happens, you might better select the protein that also has a mutation a little bit, so that that's gonna better bind to the new spectrum or new composition of the satellite DNA. So I think that's how DNA sequence and then binding proteins are kind of drifting, not necessarily actively evolving. That's what I think right now. I mean, we don't have evidence, but we think, I imagine that's what's happening.
Great segue to the next question. How does D1 protein recognize such a short cognate sequence, especially when the DNA is all coiled up within the heterochromatin?
Right. So we don't know exactly, but the D1 protein has multiple DNA binding proteins. And then this, each unit is known to go to a so-called minor group of the DNA, which is AT rich. So we probably have to do a lot more of the biochemical, analysis. I suspect, the D1 has a probably broad binding spectrum instead of AAT. For example, maybe AAT might be okay, for example. Yeah, but so how it's going to recognize exact, the sequence or some aspect of the DNA structure that, we don't have a full understanding of it yet. Yeah.
Well, I think another question is, what happens if you express the similans D1 in melanogaster?
That's a good question. We do have, the data we have been getting. Yeah. So if you just express D1 protein from similans in the context of the melanogaster, and that nothing, I mean, it looks okay. They look happy. We actually try to replace it, meaning, the melanogaster D1 mutant, that means melanogaster doesn't have its own D1 protein. And then you try to rescue that mutation by, the similars D1 gene. It doesn't work. So that's, that's pretty exciting. That means, that they are, the detailed mechanism of the function, or exactly how the protein interact with other protein and so on, is not quite the same between those two species. And I suspect that's also contribute to why, these two species cannot hybridize.
Okay, there are several questions relating to, are there any homologs of D1 proteins in mice and humans?
Yeah.
Are there any mutations in those proteins that cause diseases?
Right. So I probably should know better about the mutation. And I mean, the disease causing mutation, I don't know. We don't have, the complete, the homolog with a high sequence similarity with D1. But based on the structure, it's believed that it's the functional ortholog is so-called HMGA1. Which is also, have, AT rich bindings, the domain and so on. And then, so that, we have shown, at least in the mice cell, we haven't done anything with the human cells, but in the mouse cells, this HMGA1 has exactly the same function as D1. If you mutate, you form the micronuclei, and they are localizing to the chromosenta and so on. On top of that, you can actually even overexpress Drosophila D1 in the mouse cells, and that has the ability to bring, the chromosomes together pretty well. I mean, I don't think it can replace the function of HMG1, but D1 actually facilitates the bundling of the mouse cells.
Several more questions coming in. So there should be this one immediate follow-up from the speciation hypothesis would be that there will be a strong correlation between the emergence of these different repeat sequence and the corresponding binding protein, the D1 binding protein. What mechanism would assure this co-emergence?
Right. So, okay, this is a really out there answer. Even less, the real evidence yet. But I mean, not D1, but we have another gene with a very similar function as D1. And, how to say, they are kind of, the partner does the similar things, they're dealing with a different satellite DNA sequence, but bring all those satellite DNA together. So in that situation, we have actually the one gene that clearly goes to, the bundle, the chromosome, and then there is a gene duplication event right next to it. And then you have exact, the sister or sibling genes right next to each other. And then one is way more divergent than the other. So, and then we have a suspicion by together, these proteins, they might be, adapting to bind to, the diverging satellite DNA. Yeah.
Related question to that, is the D1 protein same across all individuals of the same species, or is it divergent within the species itself?
We don't know. I think we kind of did, again, a little bit of, the gamble sort of experiments, that we thought that's interesting. And we specifically sequenced the D1 gene from so-called balancer chromosome. Sorry that it's a little bit too technical, but that's a Drosophila chromosome that's not allowed to recombine with other chromosomes. So that means any genes in the balancer chromosome is essentially trapped. On top of that, those balancer chromosomes tend to have a little bit of different satellite DNA contents. And we thought, okay, maybe that gene might be, the D1 might be evolving. And we didn't do anything thorough, but, when we sequenced a little bit of those balancer chromosomes, the D1 didn't have any variation at all, unfortunately. Yeah.
So these repeats tend to be highly divergent. They are unstable. Do we know what's the variability across different cells within a single organism?
Yeah, that's another really good question. If you look at the just saturated DNA of the different strains, first of all, and I said the D1 protein is not different at all. But then, the target DNA sequence have a little bit of variation between the different strains of the Drosophila and melanogaster we already know. And then within the individual, I think, I think they are decently stable, but, because the destabilization tend to happen during aging. So that's that's another interesting possibility. As you get older, you might lose the the repeat number, and then next generation you might have to bounce back. So but so far we haven't detected the visible difference. But again, the minor copy number change, I don't think we can detect, for the reason we discussed earlier, that you can't just sequence, so that you cannot precisely get to the the copy number or anything. So if you lose the copy number by half, you can see it. But if you're losing like by 5%, which is substantial, in terms of the time frame of the evolution of the generation, but that 5%, I don't think we have an ability to detect that difference. So I think that's actually one major, the bump right now for us to address this specific question. So if you are interested, whoever asked this question, yeah, if you have any good idea, yeah, let me know. Yep.
Another question is, in the hybrid species, hybrid experiments, when you see these micronuclei, are there two different types of micronuclei? The chromosomes from one species go to one kind and the other go to another.
So it actually doesn't look like that way. My cartoon was admittedly a little bit, misleading, just to, conceptualize the, visualize the idea. We know when chromosome formation doesn't work, all chromosomes are affected equally, and then you just, the just ability to bundle anything is compromised, actually. So that we guess that there's some sort of, the biochemistry of the protein within a cell is making that way. So that means when we see the micronuclei, it's not necessarily any specific chromosome or any specific pattern we can discern that, yeah.
So just to clarify, because there is another question relating to it. So you don't observe, it's not the same chromosome which you always see going to the micronuclei.
Yeah. Yeah, I mean, there might be some, some pattern that we haven't recognized, but no obvious patterns like that specific chromosome always go that way, or this and that chromosome are always together or always separate. I mean, that we don't see any that kind of obvious patterns. Yeah.
There's a follow-up question to a previous question. So tandem repeats are present in bacteria that reproduce asexually. What roles will these repeats have in bacteria?
We don't know. And in case of bacteria, I mean, you don't even probably have to have a, the mechanism to bundle multiple chromosomes, right? You have essentially one circle that's a chromosome. So we don't know. I mean, that's a interesting question, but we don't know. But with that said, I would speculate, the repeat binding protein could have evolved for some other purpose at the beginning, and then, but the potentially that could have been, adapted later for, the bundling the chromosomes. Yeah. But yeah, I mean, that's really interesting speculation, but so far we don't have any information to, to make a connection between those two. Yeah.
This is more of a clarification question. When you mated the two fly species, it's only the germ cell. It looked like it's only the germ cells that die and not the entire organism. So can you elaborate on that?
Yes. So I only focused on that germ cells that's disappearing from the sterile female. And I briefly mentioned male would be lethal. So you can capture those males that's dying, that they die as a larvae, they don't come as an adult male. So that's why we just call it the dead. But you can actually go back in a timeline and then look at the much earlier stage before they actually die. Their somatic tissues, non-germline tissues are actually affected as well. Those cells die. And then if you look into it, they have ton of micronuclei. So cellular mechanism is pretty much the same across. That's what we feel. So with that said, then you wonder, right? Naturally, you have to wonder why female somatic tissue is okay. How come those females come out as adult fly, only having a problem in the germ cells? That we don't know. Why this, the cell type specific, the issues arise? That's actually the big question that we don't know. Yeah.
In the light of your work, how would you see hybrid vigor, or can you explain hybrid vigor in plants?
Yeah. That's now my new favorite question. So for the hybrid vigor to happen, first they have to be the hybridizable. You know what I mean? So the hybrid vigor is when two parts are not divergent enough, at least they are compatible. In that situation, I can imagine they can bring in all those the good sorts of things from each other. But then once it becomes completely incompatible, they can potentially bring in good things, right, from the gene. Hybrid vigor, I assume that is the contribution to the hybrid vigor is having the different genes that's coding a part. I mean, I'm imagining, right, the certain so but then the saturated DNA or this hybrid incompatibility is coming from the non-coding part. So the non-coding part has to first function. I mean, with the example of the Americana and the Britannica, right? So let's say that there's some entry that's missing from Britannica or Americana, right? If you bring both, you actually the increase in information. The one entry of whatever that was is missing from Americana, but existing in Britannica, some other was opposite way. You bring them together, you have better knowledge, right? I mean, I would say that's probably the hybrid vigor, the equivalent to the hybrid vigor. But that is only possible when you still have the basic, the rules of bundling those, the books together. So that means, the both of them comes as a bundled books, that's okay. But I would imagine, Americana comes as a physical book, Britannica comes with, the DVD. You might not be able to combine the information together. I mean, just as a, analogy. Yeah.
all the analogies in your talk and here as well are amazing. I think just one last question is what has been done in identifying the genes or this process of budding of nuclei into micronuclei? What do we know about that process? I think there was a related question earlier. Can you tie it to micronuclei in cancer or other diseases?
Oh, yeah. So the previously, the micronucleus has been mostly studied in the context of chromosome segregation. When chromosome is trying to divide and then it goes to the two daughter cells, some chromosomes are left behind in the middle. And then, when they cannot completely incorporated to the vast majority of other chromosome that forms the micronuclei. And so because of this prior knowledge, when we realized, the D1 mutant has a ton of the micronuclei, we looked into this mitotic defect a lot. I mean, seriously, because we thought that's the answer and then we didn't find that answer. And then when we did the live observation of this D1 mutant, what we saw is it's not a mitotic defect. You look at the interphase nucleus like this, and then all of a sudden, the portion of the micronuclei just indeed buds off. So the molecular mechanism of forming micronuclei seems to be quite different between what we are seeing versus in other the studied context. With that said, in related to cancer, right? So for the tumorigenesis, it's suspected that first you form the micronuclei. Inside that micronuclei, there's tons of DNA damage happens, and then that you end up is mutating some critical genes, and that causes the cancer. So that means, okay, if you disrupt chromocenter in a way that's not too severe, and then cells survive, animal survives, and then that might cause the some micronuclei to form and then mutate enough, the chromosome damage, and then mutation forms, and that leads to cancer. That's a really interesting possibility. That's one thing actually I really wish if anybody has, this must have been super easy PubMed search. I feel ashamed that I have never done that. All those, the chromosome proteins in the mammals, are they identified as oncogene or tumor suppressors? I don't know. I haven't done any thorough literature search. That's really obvious things, we should be doing. Yeah.
That's great. I think we have reached the conclusion for today's presentation. Thank you so much, Yukiko, once again for being with us here today and sharing all the wonderful insights. And thank you to all the members of the audience. Please save the date for our next webinar, which will be on June 15th. And we'll be featuring our newest invited fellow, Tobilona Oni. And until then, please take care and stay healthy. Bye-bye.